Hi guys, so this video is all about the Calvin cycle. We have spent the past couple of days in class talking about photosynthesis. There's two main reactions in photosynthesis, right? There is the light reaction and there's the light independent reaction, also known as the Calvin cycle. Remember, I don't really love the phrase light independent reaction because you need light to make the light reaction happen to give the things that are needed for the Calvin cycle to happen. So like both need light. So this video is all about the Calvin cycle. You do not need to know the Calvin cycle in quite as much detail as you need to know the light reaction. So these notes are going to be a little bit quicker than all of the time you put into your models today. So if you didn't already get one of these pieces of paper from me, because maybe you were on the Hamilton trip today, just leave yourself a space in your notes and tape it in when you get it tomorrow. Otherwise, we're going to kind of be talking about this process. This is the Calvin cycle. So you can kind of see here that there are three parts that have divided this cycle into. There's part one. See how I wrote one? This whole area, everything that's happening there is part one, phase one. There's phase two. That's everything happening in this area. And there is phase three. So that's all of this stuff, all of these reactions. Each of these phases has a name. The name of the first phase is carbon fixation. You might remember we've already talked about that phrase. Carbon. Let's zoom out and move this up. Carbon fixation. So in this video, everything that I write, you should write. So it's a little bit different than the other videos. It's more like in-class notes. So carbon fixation is when carbon from CO2 is attached to an existing 5-carbon chain called RUBP. Let's write that down and then talk about what it means. So again, carbon from CO2 is attached to an existing 5-carbon chain called RUBP or Rubisco. In the diagram here, we can get a better look at that. So one of the wonderful things about this being a video is that you can pause it, make sure you're finished writing, and then keep going, just if I'm writing too fast for you. So in this diagram, we can see that a carbon dioxide molecule is entering the cycle here. It is being attached to RUBP. That's that 5-carbon chain right here. You can see that this carbon is attached. See that circle that's now there that wasn't there before? So now it's a 6-carbon chain. So each of my little circles is a carbon, right? So 5 carbons here plus the one from CO2. Now we have a 6-carbon chain. This is pretty unstable, so it splits almost immediately into 2 3-carbon chains. Okay, so that's the carbon fixation part, the adding the carbon from CO2 to an already existing chain of carbons. Specifically in this, it's called Rubisco, the 5-carbon chain. Our next step is called reduction. So you might recall that reduction usually means adding electrons because we're reducing the charge because electrons are negative. So specifically in this process, reduction is so it's a series of reactions that adds phosphate via ATP and electrons via NADPH, forming a 3-carbon molecule called G3P. And G3P is the product of the cycle. So let's write all that down. So a series of reactions that add phosphates, and that's done by ATP, and electrons, That's done by NADPH. Forming a 3-carbon molecule. So I'll zoom in. Oops, let's see. Okay. Forming a 3-carbon molecule called, let's get another piece of paper. The lights. Okay, called G3P. This is the product of the cycle. So again, pause if you haven't finished writing. This is the product of the cycle. So if we go back to our diagram here so I can kind of show you what's going on. We're here, right? We just finished carbon fixation. We're in reduction now. So ATP is adding a phosphate. See how that P wasn't there before on that 3-carbon chain? You'll also notice that I wrote that it is made into ADP, and I didn't write a PI there. It's because that phosphate was added to that 3-carbon chain instead. Also NADPH, which has electrons, right? You know that from the light reaction. Releases its electrons, gives them to this molecule, and becomes NADPH, creating this compound, this molecule called G3P, which has 3 carbons. So if we think about it, glucose has 6 carbons, so we're going to need 2 of those to make a glucose molecule. Okay, keep that in mind. The last phase that's in red in my picture is regeneration. So let's write down what that is. So phase 3 is regeneration. So some of the G3P is used to remake Rubisco, RUBP, so that the cycle can happen again. So some of the G3P is used to remake RUBP so the cycle can happen again. You may be thinking, what do you mean some of the G3P? Didn't you just say G3P is a product? Yep, good point. So let's think for a second. Glucose has 6 carbons. How many carbons does a CO2 molecule have? Only 1, right? So this cycle has to happen multiple times to get enough carbons in here to make a glucose. So the cycle is happening, I think of it happening, I mean it happens like thousands, millions of times, but I think about it happening 6 times specifically because glucose has 6 carbons. So glucose has 6 carbons and CO2 has 1. So that's something to keep in mind. So you'll see in the next video, the TED Ed video that I'm posting, how this works. But basically, some of the G3Ps, 2 of the G3Ps are going to come out that are made from the 6 cycles and form glucose or starch or another carbohydrate component. I guess you need glucose to put together to make starch. And then some are going to be used to regenerate RUBP so that you have that 5-carbon chain again to start this cycle, because you can't do the cycle if you don't have RUBP, if you don't have the 5-carbon chain. So again, the video that you're going to watch next, the TED Ed, will have more about that whole process. It does a really good job of like making a cartoon to show you how that works, how it's regenerated. Okay, if you have questions, ask me tomorrow.